Good morning, everybody. Hope you're doing well. We're going to start today with some do now questions, like we usually do in person. I know you haven't been seeing a ton of these lately, but I'm going to start incorporating them into all of our videos so that we still get practice with Regents questions. So let's start off with our first one. So human egg cells are most similar to human sperm cells in their what? So take a second, pause the video, see if you can answer that. Right? The answer is chromosome number. Right? So remember we learned eggs have 23 chromosomes, sperm have 23 chromosomes, right? For a total of 46. So that is the only thing really that they have in common is their chromosome number and that they carry genetic information, which are the chromosomes. Everything else is not the same, right? So shape and size is of course different from a sperm and an egg. Degree of motility. Motility means movement. So sperm are able to move. They actually swim, and they're able to move on their own. Eggs are not able to move on their own. They do get released from the ovary, and then they move throughout the female reproductive system, but they do not do so on their own. They have to do that through hormones and through the menstrual cycle. So it is not on their own ability, just like sperm can do. And the amount of stored food. They don't really eat like that, so food is not a good term to use. So that is not correct. The only right answer is chromosome number. Okay, let's try another one. A pattern of reproduction and growth in a one-celled organism is shown below, right here. So which statement best describes this pattern? Take a second, pause the video, see if you can answer it. Right, the answer is A. All genetic material comes from one parent. Right, so this is cell division and growth in a unicellular organism. It is mitosis, and this is what we learned at the very beginning of the unit, where you have one mother cell that splits into two daughter cells, both containing the exact same DNA and genetic info. And then you see that this one starts to grow, this one starts to grow, but the only answer is A, because there is only one parent present here, right? Every other option here is incorrect. This is the only one that works because there is one parent, all right? So whenever you see a question like this, always look to the top and see if there is one cell or two cells. If you see two cells, one of them will likely be shaped differently, and that represents the sperm, while the circle represents the egg. And in that case, it would be sexual reproduction, so this answer would not be correct. It would be this one, right? Only some comes from one parent and some comes from the other parent. But because there's only one parent here, all of the genes, all of the genetic info has to come from it. All right, let's do one more. So in an environment that undergoes frequent change, species that reproduce sexually may have an advantage over species that reproduce asexually, because the sexual reproducing organism or species produce what? All right, take a second, pause the video. Okay, the answer is C, offspring with more variety. Remember, we discussed sexual reproduction involves a mixture of genes, right? 50% from mom, 50% from dad, and because of that, the genes mix, and we get variety. That's why none of us look alike, because humans reproduce sexually. We have a lot of variety. Also, think back to when we spoke about biodiversity. The more biodiverse, the more variety there is, the more stability it is, the better things are. So this is the only right answer here, because the others do not make sense, right? Sexually reproducing species produce more offspring in each generation. That is false, right? We know that to be false because we learned that one of the advantages of asexual reproduction is that they produce a ton of offspring, while sexual reproducing usually only a few, right? For humans, it's usually one at a time. Identical offspring, again, this represents asexual, not sexual. A new species of offspring in each generation. That doesn't make sense either, right? So whenever you're looking at sexual versus asexual, always think about the variety. Always think about how sexual is way more variety, asexual is less variety, okay? Alrighty, so Before we move on to meiosis, there are a couple of things that I would like to review. The first one is what diploid and haploid mean. Very important. So diploid means that there are two of each kind of chromosome in each cell, right? So we are diploid organisms because, again, we have 23 and 23 to give us 46. So remember, N represents the number of different chromosomes an organism has. We have 23 different chromosomes, right? So 23 times 2 gives us our 46. So think back to here. This is what we're talking about. We have 23 pairs of chromosomes. 1 pair, 2 pair, 3 pair, 4 pair, 5, all the way to 23. We have 23 pairs. But we have 46 total because we get 1 from mom and 1 from dad to give us our first pair. 1 from mom, 1 from dad to give us our second pair. So if you were to count all of these chromosomes, you would get 46, but you only have 23 pairs. So when we talk about diploid, you have to remember that it is 2n. This is how it's represented. You may see 2n on a homework question, on a test, on a quiz, on whatever. Remember, 2n means diploid. When you think of diploid, you think of 2 times 23, because we have 23 chromosomes from mom and 23 from dad. You multiply that together, you get your 46. All right, so that's diploid. Now, haploid is a little different because haploid is only 1n as opposed to 2n. So really what haploid is it's just breaking down diploid into its individual parts. So egg and sperm must have half the number of chromosomes that we need so that when added together, the zygote will have the proper number. So because the zygote, right, and remember back to our vocabulary term, zygote is when the egg and sperm fuse. Because the zygote needs 46, as a human, we need 46, we need to have 23 from the sperm and 23 from the egg. And we're going to see in a second that meiosis, right? We've learned about mitosis, but now meiosis is the way that we ensure that each egg and each sperm has 23 chromosomes and not anymore. Because if they did, when they fused, you would have too many chromosomes. And if they had too little, right, if it was under 23, then the zygote would have less than 46, which is also not good. So the meiosis process we're going to look at tells us how this happens, but I just want us to remember that haploid means 1n, right? Diploid means 2n, haploid means 1n, and the only time you're really going to worry about haploid and 1n is when you're talking about an egg cell on its own or a sperm cell on its own. So an egg is 1n, a sperm is 1n. You combine 1n and 1n to get your diploid 2n. Okay? So I know that's a lot of numbers, right? But let's just, replay it if we have to. Go back if you need to. Just remember, zygote has 46 chromosomes. Zygote is a diploid. Diploid is 2n. Sperm has 23 chromosomes. That is a haploid 1n. Egg has 23 chromosomes. That is a haploid 1 egg. 1n. So when you combine those two haploids, you get the diploid. All right? Good. So let's move on and talk about meiosis. Okay? So take a second, read this, and then I'll go through it with you. Okay. That was my way of pausing. I needed to drink. Okay, so the cells which produce eggs and the cell which produce sperm are diploid or 2n. So how do the egg and sperm cells get to be 1n? This is important to understand, right? Because we talk about how egg is 23 chromosomes haploid, sperm is 23 chromosomes haploid, but it only is like that because of meiosis. In reality, when sperm and when egg are created, they are 46 chromosomes on their own, right? So as you create an egg cell or as you create a sperm cell, you are creating them with a complete set of 46 chromosomes, but that can't be passed on, right? You can't have the egg and the sperm be 46 because if you do, then when they combined, you would have 92 in the zygote, and that is way more than you need. So the process of meiosis, right, is defined here. Meiosis is a process of reduction division, right, in which the number of chromosomes per cell is cut in half through separation. So basically, meiosis, what they do is they take a sperm cell and they take an egg cell and they take those 46 chromosomes and they cut it in half to have 23. And then once the cell goes through meiosis, it ends up with only 23 chromosomes, so that when it combines with another cell, it can produce the 46 that you need. Okay? So I'm going to repeat that one more time, and I'm going to just make sure we get that point. Meiosis is used to ensure that a sperm cell or an egg cell has 23 chromosomes in it. It's to ensure that the egg cell or sperm cell is a haploid, right? A 1N. So that when two people have sex and intend on reproducing, the sperm and the egg are each 23, so that when they combine, they become 46. If we didn't have the process of meiosis, sexual reproduction would not work, because each person would be passing on 46 chromosomes for a total of 92, which is not what we want. All right? So let's get into it and see how it's done. All right, so phases of meiosis. So a couple of things to note about meiosis before we actually get into it. The first thing, they occur only in sex cells, egg and sperm. Meiosis does not occur in any other cell in the body aside from the egg and the sperm cells. That's it. So you're not going to have body cells undergoing meiosis, right? If we get into a car accident like we discussed and we hurt our kidneys aren't going to undergo meiosis. They're going to undergo mitosis in order to replicate and fix and heal themselves. Only egg and sperm undergo meiosis, right? And again, the purpose is to reduce the number of chromosomes by half. Okay? So, some important things right here. Meiosis, just like mitosis, is preceded by the replication of chromosomes. All right? That is important. Part 1 right here. Meiosis, like mitosis, is preceded by the replication of chromosomes. Okay? So, what that means is when we went through mitosis, we remembered that the first part, before mitosis takes place, we have to go through interphase. Specifically, which part of interphase, right? I'm asking you. I know you can't answer me, but think about it yourself. Make sure you remember it's S phase, right? S is when the DNA and the chromosomes get replicated. It happens before mitosis. In meiosis, the same thing is true. The DNA needs to replicate before it goes through meiosis. Okay? The only difference is that replication is then followed by 2 divisions. There's meiosis 1 and meiosis 2. So we're going to see that in a second, that is the main differentiation between meiosis and mitosis, is there's 2 divisions. You're going to see the names of them are the same. The stuff that occurs in them are also the same, but there's 2 separate divisions. There's a meiosis 1 division and a meiosis 2 division. All right? So that's going to make more sense on the next slides when we actually see it. Okay? So, meiosis 1. This is the first stage of meiosis. It's 4 stages. All right? So meiosis 1 involves interphase. This is where the chromosomes replicate. Remember, just like in mitosis, interphase encompasses G1, S, and G2. That is the same for meiosis. Interphase, G1, S, G2, the same exact stuff happens, especially in the S phase. The chromosomes replicate, right? It is similar to the chromosome replication of mitosis. 2 identical sister chromatids are held together. So remember, the way it looks the same too. This is the same. Now I'm going to just tell you that real quick. You don't really need to worry about how to identify the pictures in meiosis. I would worry about identification just for mitosis. For meiosis 1 and meiosis 2, just remember what occurs in each. So the beginning of meiosis 1 is interphase. Just like in mitosis, interphase involves the replication of chromosomes, and it involves the growth of the organelles and the growth of the cell. Then you get to prophase 1, right? Prophase 1, chromosomes will shorten and thicken. Each chromosome pairs with its corresponding homologous chromosome. Remember, homologous means the same, right? to form a tetrad, right? A tetrad means 4, right? So there are 4 chromatids in a tetrad. And if you look at this picture here, you'll see 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4. So tetrads form in meiosis. Basically, what this just means is that you have 4 chromosomes that link together, right? You have the one here, you have the one here, 1 and 1, and it ends up being 4 of them, okay? So that's really all you need to remember about prophase. Metaphase, the tetrads will line up in the center of the cell, just like it does in metaphase in mitosis, right? Metaphase 1. And then anaphase 1, the tetrads will break and move to the opposite sides of the cell, just like it does in anaphase in mitosis, right? That right here is meiosis 1. Now, a couple of things to remember about meiosis in general. Notice how prophase, metaphase, anaphase, they have a 1 next to it. That is incredibly important because remember, where in meiosis 1, there is a meiosis 2 that we're going to get to. It's also incredibly important because if you didn't put the 1 here, then it would just be prophase, metaphase, anaphase. That's the same as mitosis, right? And you can't have that. There needs to be some sort of differentiation between mitosis and meiosis. It becomes difficult because the names of the phases are the same, as you see, right? Interphase, prophase, metaphase, anaphase, that is the same as mitosis. The way that we differentiate, the way that scientists differentiate, is by placing the 1 after prophase, metaphase, and anaphase, and by realizing that in meiosis, there's a tetrad, whereas in mitosis, there isn't. Okay? So that's meiosis 1. Meiosis 2 involves telophase 1, right? So this is still, technically this is still part of meiosis 1. We just couldn't fit it on this slide here. So technically, I'm going to say that again, telophase 1 is part of meiosis 1. So just like in mitosis, you have interphase, prophase, metaphase, anaphase, telophase. The same is true for meiosis. You have meiosis 1, starting with interphase, replication of DNA. You have prophase 1, where the tetrads are formed. You have metaphase 1, the tetrads line up in the middle. You have anaphase 1, where the tetrads split and move to opposite sides of the cell. And then you have telophase 1 and cytokinesis, where what is this called? Do we all remember what this is called? The indent here? It's called the cleavage furrow, right? You have the cleavage furrow that forms, and you end up with a split. And now you have 2 distinct cells right here. Now, the other differentiating thing between meiosis and mitosis is now that the cell has split, right? The cleavage furrow has split the cell into 2. You now need to go through another stage of meiosis, right? If this were mitosis, we would be done because we've now created 2 cells from 1. But in meiosis, you need to continue and you need to split the cell again, right? So what do? We go through meiosis 2. So again, it's the same exact names, except now there's a 2 next to it because we need to differentiate 1 from 2. So we have prophase 2, where you have the sister chromatids. They start to move towards the center, just like they did in prophase 1. You have metaphase 2, where the sister chromatids line up in the middle, just like they did in metaphase 1. You have anaphase 2, where they start to pull and move to opposite sides of the cell, just like in anaphase 1. And then telophase 2, where the cleavage furrows are formed again, and they are now splitting. And it results in 4 cells, 1, 2, 3, 4, that are all 1N. This is the key here. The only way that we get these cells to be haploid, right, which we talked about here as the entire purpose of meiosis is to reduce the chromosome number. The only way we do that is if we go through a second stage of meiosis to get them to be 1N. All right? So this is meiosis. That's it. It's really not that complicated. I'm going to review some key points right now. All right? The key points I'm going to go over are as follows. Meiosis 1 and Meiosis 2 are essentially the same thing. It just happens 2 times. The stuff that occurs in Meiosis 1 occurs in Meiosis 2. It's just the same thing happening again. All right? Remember that you need to differentiate prophase 1 from prophase 2. You have to actually write the number 1 and 2. The same of metaphase, anaphase, and telophase. Okay? That's one other thing. The other thing is when you were to go through meiosis 1, right? So you have duplication, you have replication, and then you split here. This cell is a diploid, right? Each one of these is a diploid. And we remember that meiosis the whole purpose is to split it again so that we reduce the number of chromosomes. That's why we go through this process one more time to get 4 cells that are all 1N or haploid. All right? And that's pretty much it. So for meiosis, again, remember what happens in each phase. Do not worry about how to identify them. Just know what happens. Make sure you understand what a tetrad is and why there's a tetrad, right? The reason there's a tetrad is because there needs to be one of them in each of the 4 that we end up producing, right? 1, 2, 3, 4. There needs to be one of these in each of those, right? And then just remember that meiosis 2 occurs because you need to further split these cells and make them haploid. Okay? I realize that this is a lot. We're going to finish off the slides. There's not a whole lot left. I understand that this is, hopefully not too confusing, but that you may have some questions. So remember, as always, you can always reach out, post questions in the question thread, email us directly, whatever you need to do. Just make sure you're reviewing this. All right. So the importance of meiosis. As we said before, the whole reason we do meiosis is to turn one diploid cell into 4 haploid cells. 4 of them. We need 4, right? So the chromosome number of the egg and sperm is cut in half to ensure, that should be an E, to ensure that the zygote will have the proper number of chromosomes. Remember, we need to reduce the amount of chromosomes to 23 in order to ensure that when they fuse, we get a proper zygote with 46 and not more. Okay. So, crossing over. There's an idea of crossing over that's very important. This is a very simple thing to understand, but you got to really get it. Crossing over means, right, that chromosomes will actually cross with one another. Notice here how you have a red chromosome and a blue. Eventually, you're going to have a red chromosome that has part of blue and a blue chromosome that has part of red. This idea of crossing over means that there is an exchange of genetic information between segments of chromosomes during meiosis. There is an exchange of genetic information. All right? This happens during prophase when it lines up next to its homologue. This is how we get the tetrad that we were speaking about before. Tetrads will cross over. So the whole idea of crossing over, what I need you to understand about it is I'm not super concerned with you knowing when it happens. I'm more concerned with you knowing why it happens. And the reason that it happens is because this is how we get genetic variability, right? There is an exchange of DNA, of genes. So that means that this chromosome right now is no longer the same chromosome it was before. It has DNA from a different chromosome. So it exchanges some information and it becomes different. And this is one of the ways that we end up becoming different and variable ourselves, right? Because you now have a mixture of DNA so that when they combine the egg and the sperm, there's even more of a mixture, right? This all has to do with variability, and it has to do with, why we would want to do this, right? We want to make sure that we're variable for many reasons that we're going to talk about in a bit. But I'm concerned with you understanding that crossing over is when homologous chromosomes exchange genetic information during meiosis, and they do so that they can increase the variability. They can change their genes, and they can become different, so that there is a different gene or DNA segment present. All right? That's what I'm concerned with you understanding. All right? So that's what we need to remember about crossing over. It's an important concept, but we're not going to get into it too much now. We talk a little bit more about this in our DNA and genetics unit, but for now, just remember what we spoke about. Okay? All right. So this is basically what we just discussed. So the result is that the offspring will receive a new combination of genetic information. This leads to variation in the offspring. So this is all stuff that we spoke about. I'm not going to read through all of this right now, just in the interest of time, but please pause the video here and just fill in your guided notes or take notes on this information here. This is everything that we already discussed about variation and sexual reproduction, but just so you have it on your notes. All right, so pause and fill this in. Okay. All right. So meiosis produces 4 haploid cells that are different, right? In males, meiosis results in 4 sperm cells. So every time one sperm cell that we create undergoes meiosis, it creates 4 more sperm cells. In females, the same thing happens. 4 cells are produced, but only one of them will become an actual egg cell, right? All of the cytoplasm and the organelles that are reproduced and replicated during interphase, right? So we know that happens in interphase during G1. So all of that only ends up being put into one of the 4 egg cells that are produced, where in males, you get 4 sperm cells that are all functional, right? This here is an important thing to understand. This is why men are able to have children for pretty much their entire lives, right? There really isn't an age where a man is not fertile anymore, unless, of course, there is an issue with genes or DNA, or there is another genetic problem that prevents him from reproducing well. But for the most part, most men are able to reproduce for their entire lives. This is also the reason why most females, or I should say all females, are not, right? There is a certain point in a woman's life where they will no longer be able to have children. And that is because women produce way less eggs than we do, than we produce sperm. Way less, right? Women produce way less eggs. And this is one of the reasons that during meiosis, you're really only getting 1 functional egg cell out of the 4 that you're creating. So women produce eggs at a much slower and much more infrequent rate, and that's why they are able to have children only up until a certain point. And then at a certain point, egg cell production stops completely, whereas that does not happen with sperm. Okay? So we're going to finish off with a quick comparison of mitosis and meiosis. Mitosis occurs in all cells of the body except for the egg and sperm, while meiosis only occurs in egg and sperm. One big difference. Another big difference. In meiosis, each diploid cell divides twice to produce a total of 4 cells. In mitosis, each diploid cell divides only 1 time to form 2 cells. That's another big difference. In meiosis, each of the 4 cells contain only half the number of chromosomes as the parent, while in mitosis, each new cell contains the same number of chromosomes. In mitosis, the homologous pairs do not come together to form tetrads, but in meiosis, they do come together to form tetrads. And then, while they're in tetrads, crossing over may occur. There is no crossing over in mitosis because all the DNA stays the same. There's only a change in DNA during meiosis. And last but not least, in meiosis, the 4 haploid cells contain different combinations of chromosomes, right? And in mitosis, the new cell contains identical copies of genes. All right, guys, so we are done with meiosis as far as the PowerPoint slides are concerned. The next couple of days will be some articles that we're going to read that will help solidify this information. There'll be a review, there'll be some multiple choice and fill-in and writing questions. But as far as the actual PowerPoint slides are concerned, we are done with meiosis and mitosis. So you should be done with the guided notes at this point. Make sure you use that to study. And as always, please ask if you have any questions about this stuff. All right, so we're not done with this unit yet, but we are done with the PowerPoints. So we'll get to the rest of it tomorrow. All right, have a great day, and I'll talk to you soon.